here that cut down the hydrogen bond and separate the two DNA strands together. We have the stabilized protein that keep the two strands that not come back and bind together. And we have primates, which will add our RNA primer into the template. The DNA polymerase will start to synthesize the newly DNA strand from the position of the RNA template. We have the nuclease, which will delete the RNA primer and replace it with the DNA base. And finally, we have the ligase, which seal the sugar and phosphate backbone and seals the discontinuous DNA strand together. And as a consequences from one strand, double strand of mother DNA, we will have two daughter strand of double strand DNA with the sequence relatively more or less quite the same. And during the cell cycle, we have forming phase, the G1s and G2 phase is the interphase, which is also as a longest phase in the cell cycle. And we have a mitotic phase, which can categorize into the prophase, metaphase, anaphase, and telophase. And come along with the cell cycle checkpoint, we have three main cell cycle checkpoints. One is in the end of G1 phase to checking for if the DNA was ready to the DNA's replication with all the necessary enzyme, and the DNA was fine without any damage. The G2 checkpoint, which check for all the process of the organelle DNA was replicate smoothly, prepare for the mitotic phase, and the spindle checkpoint or metaphase checkpoint in the end of metaphase to check for if the chromosome was separatedly directly into different side of the cell. If the process going through each checkpoint, it will maintain the cell was divided accordingly to the signal of the cells. So first, let's watch again the video demonstrate how the mitosis were happening and what is function of each cell cycle checkpoint. What's wrong with this? Okay, so okay, thank you very much. Sorry for the technical problems. Okay, share. Okay.
An adult human contains an estimated one.
Is it okay now? Everyone can hear clearly? Is it okay for everyone?
100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or gap 1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S, or synthesis phase, the DNA replicates. Finally, during G2, or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1s checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
So, for the cell cycle, we know that in the zygote, the first stem cell, when the sperm combine with an egg, have a cell cycle about 24 hours. So that means that after 24 hours, one day, it will divide from one zygote into 2 cells. And after another day, it will divide into 4 cells, another day into 8 cells. So come with the differentiation, the zygotes will develop into different cell type, and each cell type will have different cell cycle period of time. So in all the human cell, the duration of the S phase, the G2 phase, and the M phase are more or less quite the same. The total of these 3 phases is taking about 12 hours. However, the most variable phase is the G1 phase, which quite different in different cell lines. For example, I have provided for you in here a table with the cell cycle duration of time of different cell types. So, for example, in skin cell, we have the cell cycle taking about 2 weeks. In erythrocyte, the red blood cell, the duration of lifespan is around 4 months. The liver cells is about a year. Intestine in the gut, cell in the gut is about 4 to 5 days, and especially the muscle cell is taking about 16 years. And there's some hypothesis that why we rarely have a muscle cancer or heart cancer. Yeah. So we have all type cancer. So in Vietnam, the most popular cancer, for example, in human, we have the most popular cancer is liver cancer, lung cancer, and prostate cancer. And in women, we have the most popular cancer is the ovarian cancer and the breast cancer. But we rarely have the heart cancer or muscle cancer. So there's several hypotheses on this, on why we rarely have this type of cancer. First of all, we have the hypothesis is the muscle, and especially the heart, is not the organelle that contact with many mutations factor. Like in the other organelle, like in the lung. The lung is where the air, the blood carry the oxygen and carbon dioxide we expose with the air. So if you have breathed some polluted air, it will have harmful with the lung cell, and it will activate some DNA mutation. And if the DNA mutation was in the critical position, it can bring cancer. Or the intestinal system, the cell in the digestive system, to extract the nutrient and other metabolic mechanism. If you eat some bad food, it can activate some mutation. However, the muscle, cell tissue, and the heart is contact with none of the potential mutagenesis agent. So this is the first hypothesis why we rarely have the heart or the muscle cancer. But the other hypothesis is due to the very long duration of the muscle cell. So in the last session, we have talking about the DNA mutation. And we know that, in human, the mutation rates normally without the DNA repair mechanism is about 1 to time 10 power minus 8. That means in 100 million base pair we'll have one mutation, and due to the DNA mutation repair mechanism, it will decrease this into 10 billion base pair. That means in around 3 generations of cell cycle, because the human genome is about 3 billion base pair. So more than 3 cell cycle will have one mutation. And the long lifespan, the long cell cycle, that means the less the cell have to divide, and the less it have to divide, the less chain DNA mutation will occur in this kind of cell type. Okay? And this might be a very hypothesis to explain why we rarely have the heart or the muscle cancer. So talking about the development in cancer. So cancer is start from DNA mutation. So this DNA mutation causing the cell divide uncontrollably. With a normal cell, it must divide when it receive a signal, a cytokine of the body tell the cell to divide. Under certain circumstances, for example, it needs some new cell to replace the old one or for development. But for the cancer cell, it divides even when it did not receive any signal from the body to tell it to divide. So it divides uncontrollably, and when it's divide and keeping divide, it create a tumor, a cancer tumor. And when the cancer tumor becomes too big, it affects to the effective of the other organelles located beside of the tumor. And when the cancer cell detached from the tumor to follow the bloodstream, going to the other corner, other organelles of the body, we have the malignant state. So in the malignant state, there's let's say that all the treatment, all the therapy, all the chemical agent are not really effective, because we have the cancer cell in every corner of our body. Okay? It follows the bloodstream to travel to everywhere and create a tumor developing there. So it's really not effective. At what stage people can check the cancer cell? So it really depends on which kind of cancer. So for example, some type of cancer really really hard to detect. For example, in the liver cancer, we just know there's some polyp there, but we don't know that if that is a normal polyp, that means a small tumor and it not develop anymore, or it actually cancer. Some type cancer are more easier to detect. For example, the intestine cancer or the colorectal cancer. It really depends on the type of cancer. But mostly people detect the cancer cell when they have a relatively health check, and normally they will found it in the late of the stage 2 and in the beginning of stage 3 cancer, most of the cancer. But it really depends on the type of cancer. Some type cancer are really hard to diagnose. So according according to a paper was published on 2009, that states that we all have a chance to develop cancer cells in our body. And the more age you have, I mean, especially for the people who over than 40 and 45 years, 45 years old, there's definitely there cancer cell developing in our body. However, this cancer cell cannot develop into the cancer tumor because of our immune system recognize the cancer cell and recognize this is the cell that divide uncontrollably, and our immune system kind of attack to this kind of cell and prevent it to develop into the tumor. But sometimes the immune system doesn't work and we still have cancer. So yeah, I still have a question here. How can we know the melanoma in our body? It depends on the we want to know it deeper. Melanoma is a first type of cancer, skin cancer cell. So what kind of melanoma you want to know more?
Teacher, can you hear me?
Yeah.
I think I will pick honestly, I really don't know about the melanoma too much because I have seen it through my grandma's cancer, and I know it. But I know it depends on the immune system. And by the way, my grandma has a lung cancer.
Okay.
And unfortunately, she has gone 3 years ago.
Okay, sorry for that. And
Yeah.
your question is, how can we categorize from a normal tumor with a cancer? That's what you mean?
Yeah.
Okay, so a normal tumor like a polyps. So everyone will, at a certain age, we can develop a normal tumor. But I think that's two gold standard to detect that if that is a normal tumor or a cancer. First, it is developing inside. For example, if we see a tumor somewhere and we see that it's larger every day, it's not a good signal. And then we will have to go to the hospital to check it. They will develop a test on the cell. They will pick up some of your cell and to put it in the malignant state to check that if this type of cell can develop a malignant state. And if it can develop a malignant state, it's definitely a cancer cell. Okay. So, all right. Am I answer your question or not?
Yes, thank you, teacher.
Okay. So another very interesting research performed on the cancer, just a skin cancer patient. So normally, if you have cancer without any treatment, you will be killed in after a certain years, depend on the cancer. However, a research conduct in a group of patients has found out that there is several cases of patients somehow they overcome cancer without any treatment. So when the scientists further investigate on those patients, because it's really strange if you get out cancer without any treatment, they find out that in those patients, during they getting cancer, they also getting infectious disease. That means the infectious agent could be a virus or bacteria or fungi or whatever. And during they get infectious disease, of course, their immune system will reply. They will have a fever, and the fever actually is a good thing. The fever is a signal of the body to activate the immune system, to activate the white blood cell, to make it stronger, and have open their radar to detect more, let's say, a dangerous factor. And when this white blood cell was activated and defeat the infectious agent, they find out that they attack also the cancer tumor too. And after the infectious disease was gone, the size of the cancer tumor was decreased significantly. And it leads to nowadays very trendy therapy of to treat cancer, which is the immunotherapy to treating cancer. Any question? One more thing is interesting about this study is they also investigate the effect of paracetamol on the treatment, because you know paracetamol is to calm down the fever. If you have the fever is more than 40 degrees C, something like that, normally the paracetamol will calm down the fever really quick and make you easier to suffer from the fever. But they find out that with the patient who getting both cancer and infectious disease, when they overuse the paracetamol, normally the fever become mild, but the cancer cell was not decreased. The cancer tumor do not decrease. So it means that the paracetamol have, besides of calm down the fever, it also make the immune system weaker to detect of the outsider to attack the cancer cell and the infectious factor. It's a quite interesting research.
Can I have a question?
Yeah.
I don't know, it may sound a little bit stupid, but what happens if all human cells are cancer cells? Would we have a horrible death, or humans would evolve into a more supreme kind?
Well, normally all the cells.
Yeah, I mean, most mutations are harmful. Like, it's not neutral.
Okay, so your answer for the question for him or what? Okay, so normally all the type of the cell in the human body must divide following program. So for example, if you are a child like 4 or 5 years old, also must be divided much more than the old people who are around 60 years old. So each cell line must divide following when the body tells it to divide. And if a group of cells or all the cells in the body, like the case you asked me, divide not following the exactly program, there is a problem. So not every time cell division is pretty good. Okay.
May I have a question, please?
Yeah.
So you have discussed about the ways to cure cancer when a patient is suffer from other illness such as virus, right? So I think that is it possible that this will only cure the cancer when it's in the early stages, or this can cure any stages of cancer? When you get sick from a virus.
Okay. So first, this is not my study. I just read it from a paper, And in the paper, what a pity that they are not mentioned about the states of the patient. So I don't know that if the patient they are treating in here is all involved in all stages of cancer or just the people who are very early age of cancer. But the fact is, I don't know. I don't have this kind of information. Okay.
Okay, thank you.
So we have studied about cancer like more than 50 years ago, and we see that more and more genes are found out related to cancer. And the most popular genes that have been related to cancer is the BRCA1 and 2. So this gene, the mutation on this gene have been indicated associated with many type cancer, both in men and women. Like in men, we have this mutation in BRCA1 and 2 genes related to breast cancer, prostate cancer, pancreas cancer, and in women, ovary cancer and other type cancer. And yes, you are not have a problem with your eyes. In men, we have the breast cancer too. Okay, not only in women. So be careful, guys. If one day you found something weird in your breast, so please check it. Although man breast cancer is the ratio is quite low compared to the ratio of having breast cancer in women, but man can suffer from breast cancer too. Okay. Okay, so further investigate of the function of BRCA1 and 2, the scientists find out that this was encoding for the cytokine, which played a role in the cell cycle checkpoint. So it's because it played a role in the cell cycle checkpoint, critical role in cell cycle checkpoint. So if we have any kind of mutation on the BRCA1 and 2, so the more protein it encodes for will lead it to the decreased effectiveness in DNA repair and controlling the cell cycle checkpoint, especially the spindle, that means the AMP checkpoint. And it leading to the cell will divide uncontrollably because the checkpoint has been broken. Okay. Okay, so I have a question for you. So in the G1 phase of the cell cycle, primarily involve which state of the following? 1 is prepare DNA for replication, 2 is DNA replication, and 3 prepare for mitosis. Okay. Okay, so many of you have the answer is 1. And the correct answer is yes, is the 1. So the G1 phase is involved in prepare DNA for replication. And DNA replication is the phase as we have discussed earlier. Prepare for mitosis is the whole interface involving the G1, S, and G2. Okay. So another question for you. So you are looking at the diagram of the mitosis process. So please put a suitable word in the space for me. So what is this organelle? What is this? What is the state of each? What is the name of each stage in the mitosis system. Okay. Please help me to put a suitable term in the blanks for me, please, from blank number 1 to number 9. So what is number 1? Number 1 here, we provide microtubule to drag the double.
Okay.
Yeah, 1 is essential. Yeah, great. 2 is DNA or chromosome. It's the same.
Chromatin.
Yeah, chromatin. Okay. The number 3 is.
Number 3 is spindle.
Spindle or microtubule. It's the same. Okay. 4 is interphase, right? 5 prophase, 6 metaphase, 7 anaphase. Okay. The 8 is. Well, actually, 8 is the final step, which is telophase. And okay, and the 9 is cell divide. Okay.
Or is it cytokinesis?
Cytokinesis is more suitable with the 9. The 8 is
the
final.
I mean the 9.
Yeah,
I mean
the 9
the
is
9.
Yeah, the 9 you can type cytokinesis or cell division. Is we can use both. Okay. Okay, so next we will come to another process called meiosis. So meiosis we study in high school, which is occur mainly in the gamete producing cell. So from one mother cell, during it had been through one time the DNA was double or DNA was replicate, and 2 times the cell was divided, and as a consequence, it would provide 4 daughter cells with the number of chromosomes is decreased by half. And when the gamete combine together through a process called fertilization, the number of chromosomes will be restored, and as a consequence, the new offspring will have a half of the chromosome set is from mother, or sometimes we call maternal, and a half it comes from the father, or we call paternal. So this is a diagram of the meiosis. It's including 2 times of division. The first time is called meiosis 1, and the second time is called meiosis 2. So on prophase 1, the nucleus membrane was disappear. The 2 centriole was gathered in the 2 sides of the cell, and different with the metaphase. Different with the metaphase of the mitosis. In metaphase 1, the double chromosome will gather through line in the metaphase plate and the a quarter of the cell. And in metaphase 1, there is interesting phenomenon occur, which is called the crosslink. That means a part of the chromosome, which is marked in red here, exchange with the sister, with the homologous chromosome in the homologous region. So as a result, we will have a part of the double chromosome here, bring another, the homologous part from the blue one here. So in the end of telophase, in the end of meiosis 1, we will have two cells with the number of chromosome is decreased, but each chromosome is a double chromosome, okay? And in the end of meiosis 1, I mean telophase 1, also the beginning of the meiosis 2, of the prophase 2. So the membrane another time disappear, and the meiosis 2 is occur the same with the mitosis. So the double chromosome was together in the equator called the metaphase plate, and it's split under the control of the centriole through the microtubule. It's split into the two direction of the cell, and in the end, we have 4 other cells with the number of chromosomes decreased by a half, and some of those, due to the crosslink, will have a new recombinant chromosome, different with the chromosome of the parent. So let's watch a small clip demonstration about meiosis.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the
Are you hearing the, can you hear the sound of the clip?
Yes.
Very clear.
Okay.
process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centriole
Teacher, I think the vid is stop.
happened? Oh. Hello, everyone hear me? What's wrong?
Yes, I can hear you.
Yes.
Okay, so.
Have you ever wondered why children do not look exactly like their parents, or why?
So you can hear the clip, right?
Yes. Yeah.
Okay, so
Have you ever cell enters prophase 1 of meiosis? Up to this point, the cell looks similar to mitosis, but two events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Okay. So, due to the independent assortment, so a couple in human, a couple can have 2 powered by 23 chain to have a combination of one gamete. So that means that in father, for example, if you have a sibling and you take one chromosome from your father, so maybe your sibling, your brother or sister, or can inherit the other chromosome from your father. So it's come to the number of recombinant genetic background will become to 2 times 2 powered by 23. That means 2 powered by 24 change of the genetic background. So that is very large change. That's explained why we rarely have a normal sibling that have their identical genetic background together, except for the identical twins, which we will explain later. So come back to the mitosis and meiosis. Mitosis is divide in the most frequent divide in the somatic cell and also in the primitive gamete cell. So from one parent cell provide two daughter cell with the genetic the genome as the same. Meanwhile, the meiosis, the main difference between the meiosis and mitosis is in meiosis parent cell, after 2 times the cell will divide, will provide the 4 daughter cell with the number of chromosomes was decreased by 1/2, and due to crosslink, it will bring even more change of recombinant genetic background. This variation will provide material for the evolution that we discussed in chapter 4. So I had made this comparison between mitosis and meiosis for you, and as I remember, is this content is one of the survival kit, so try to remember it. So in mitosis, we including 1 time the cell was one time the DNA would duplicate and 1 time the cell was divide. And meiosis, 1 time the DNA duplicate, but 2 times the cell was divide. Mitosis appear mostly in somatic cell and in early gamete cell, and meiosis is produced gamete cell. So the consequences from 1 parent cell to 2 daughter cell, 1 parent cell to 4 daughter cell. The daughter cell will have the genetic background the same with the parent cell, and in here the daughter cell will have the genetic background first decreased by 1/2, and second may be different due to the crossing over. Okay. One more thing is the differences in the male producing gamete cell and the female produced gamete cell. So in male, male produced gamete cell will produce from one cell will produce 4 sperm. That means female gamete, because the male gamete, because the male gamete, the sperm, doesn't require many cytoplasm to survive. But in the female gamete, one time the cell divide, it will produce the unbalanced, unequal to outside here. We can see that a big one is bigger with more cytoplasm, more mitochondria, and the other is smaller. And this one will divide also unbalanced to produce only one female gamete. So in female gamete produce cell, into meiosis can produce only one functional female gamete, and the other 3 gametes was discarded. Okay. So now, before ending this chapter, I have a question for you. Please select the true statement about meiosis, and note that there might be more than one true answer. Which is of the following statement is true about meiosis? Okay, we have one student choose the number 1 and 3. How about the other? Okay, so some students choose 1, 2, 3, some students choose 1, 3, 4. Okay, so we'll see. So, okay, I see your answer. So let's come to the first statement. Meiosis involves up to sequential cell division. We'll see that in here, meiosis involved one time the DNA was duplicate, and 2 times the cell will divide in meiosis 1 and meiosis 2, right? So 1 is true statement. True. I will place my pointer, and so this is true. So second, no DNA replication occur before meiosis because the chromosome set is half. So in meiosis, it including one time the DNA was replicate. Remember? So this one is false. Crossing over leading to variation in genetic material, of course, is true, because it will provide many types of gamete due to the crossing over. It could bring the gamete with the parental chromosome, or it can bring a gamete with the recombinant chromosome. So this is true. And last one, during meiosis, paternal and maternal chromosome are separate, ending up into 2 different cells.
So they are from one, like the term are false.
I'm sorry, what?
Yeah, I think the term is false.
The term is false. Yeah, exactly, because as the clip, you can see that in meiosis, the paternal and maternal chromosome, that means the chromosome come from the father and mother, was independently assortment. That means that maybe this one is from paternal, and the other one is from maternal, is distributed independently. So totally randomly, so we don't know if we got the one from partner or from the matron. So it's ending up randomly distributed. So this is false. Okay. So the true statement is the number 1 and number 3. So we have finished the chapter 2, and now I suggest that we will have a small break, 5-minute break before we can continue with the chapter 3. 5-minute break. Okay, so let's come to our next chapter. Let's discuss about transcription and translation. So transcription, in fact, is just transfer the information from the information writing in DNA code to RNA code. Actually, the code of DNA and RNA are quite similar. Translation is a little bit more complicated since it has to transfer the information from nucleotide to amino acids. So, but the end of this whole process is to produce protein, and protein is really important. Protein is play a critical role in almost all the process inside the body. The protein could be a structural protein which can build cell. Protein could be an enzyme which is the catalyst for the biochemical reaction inside the body. The protein could be a cytokine or receptor, play a role in the communicating between cells. Protein also can play a role as a storage to store the necessary component for the body in the certain circumstances. So first, let's watch a small video clip on what is protein, what is function, and how the structure of protein can maintain its function. Okay.
Proteins play countless roles throughout the biological world. Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules. Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas in the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines at the RCSB Protein Databank.
So if you're interested in any type of protein, you can go on the Protein Databank, PDB. It's related to you to the Protein Bank. So, and like the NCBI is a data bank for genome, the RCSB is the data bank for protein. So in here, all type of the protein that humans have been study, the whole the 3D structure and the ribbons, the amino acid sequences. Let's say, for example, I want to looking for TNF here. So you can click on the protein we can here is the tree structure of each pattern of the TNF protein. Okay, and also you can retrieve all the X-ray crystal structure and the amino sequences and what is functional protein and so on. Okay. Okay, so come back to our slide, transcription. So DNA is the process transfer DNA to RNA is called transcription, and transfer RNA to protein is translation. And the protein here is just first degree, which is just a sequence of amino acid. It will have to been through a process called post-translation modification to create its PD shape to get into its function as a protein. So first, let's watch an overview on DNA structure, function of DNA to protein.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So just a quick reminder in the structural difference in the DNA and RNA, we can see that the DNA is deoxyribose, that means the 5-carbon sugar, but deoxy, that means it's lacking 1 oxygen in the position of carbon number 2 here. And in RNA is the hydrogen group attached to this carbon. And in DNA we have 4 nucleotide is A, G, C, and T. A for adenine, G is guanine, C is cytosine, T is thymine. And instead of T in RNA, we have uracil, and the only differences in uracil and thymine is just in the carbon also number 2 here, the hydrogen atom attaching to carbon instead of the methyl group. Okay. So how transcription and translation happening in prokaryotic, and what is the differences between of this process between prokaryotic and eukaryotic cell? So two main differences. The first one is in prokaryotic, because there's no nucleus membrane to separate the nucleus region with the cytoplasm. So as soon as the RNA polymerase come binding and start to synthesize the mRNA, the ribosome will come also and attach into the mRNA and start to synthesize the polypeptide. So that in here, the transcription and translation is a continuous process because it's happened nearly immediately at the same time. One other difference is the ribosome in prokaryotic cell is smaller. The ribosome was about 70S, which S is the unit for the centrifuge fork unit. Its name followed the scientist who invented this unit. And this 70S was composed by two subunits. One is have a size of 30S, and the other have a size of 50S. Meanwhile, in the eukaryotic cell, because in eukaryotic cell we have the nucleus membrane separate the nucleus region and all the DNA inside with the cytoplasm. So the transcription was happening inside the nucleus region, inside the nucleus membrane. And after the mRNA was synthesized and had been through to the mature mRNA processing that we will discuss later, it will getting out of the nucleus region through the nucleus membrane hole. And after that, the translation starts. I mean, the ribosome will come and binding into the mRNA and start to synthesize the polypeptide. So in eukaryotic cell, the transcription and translation is not occur continuously. The translation happening after the mRNA have been going out of the nucleus region. And the size of the ribosome is larger, which is 80S, consists of two subunits. The small is 40S and the large is 60S. A little bit larger than the ribosome in prokaryotic cell. So let's go further on the transcription in both prokaryotic cell and eukaryotic cell. So in eukaryotic cell, we have a typical promoter of the prokaryotic cell is come as follows. So let's hypothesize this is the position +1. This is position +1. +1, that means that this is the position that the mRNA will start to synthesize. Okay. So before this position 10 nucleotide, we'll have the region called the Pinot box. We have the compose, the sequence TATAAT. And we have another region is at the position -35. That means 35 nucleotide from this position is the TTGACA box. So how we can, how the scientists can identify which is the favorite promoter binding region of genes. So it's based on the motif binding. Bindings. Okay. So in here you can see a motif binding. of a transcription factor that they have sequence in so many many positions of the gene. So, for example, identify big binding transcription factor. So first, they put it in the genome. So this is the genome of organism. This is, let's say, the genome. And they let the transcription factor binding into it. So the red one here is a transcription factor, for example, it binding in the favorite region. So the red one is the after the transcription factor binding here, they break down the DNA sequence and start to sequence those sequences. So for example, if we have a motif binding here, is that because in many times they had find out that in the position of 3, here 100% is a T. Also in 4, in C here is, and the A in the position here. And the other, for example, in the position of the 9 here, 50%, they see that is an A, and 50% they find out it's a C. So this person in this position is either A or C, or in the other case, the transcription factor will not binding. But in this position, 3, 4, 6, and 10, if the nucleotide would change, that means transcription factor will not binding in these sequences. And that's how we can identify which is the region that a transcription factor binding in. Okay. So come back to our slide. So in here we have a typical prokaryotic promoter here. We have 2 regions that favorite for the transcription factor binding, the Pinot box and the -35 box. The typical eukaryotic promoter is from the TATA box, which is in the -35, sorry, 25 of the transcription starting site. CAAT box in -80, and one region really interesting is called the GC island. So GC island is a region that contains many G and C nucleotides, and this GC island is related to the DNA methylation that we will discuss further on chapter 7 in the epigenetic chapter. Just for now, just remember that if the gene was mostly DNA methylated, that means the genes will be turned off during transcription, mostly. So the state of transcription start when the transcription factor among those DNA polymerase will come and binding in the region and start to synthesize the RNA polymerase. And it will start synthesize the mRNA along of the length of the gene until it gets to the termination. Then it will release the DNA template and also the RNA product out and finish the transcription process.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex, able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors called coactivators link the basal factors with a third set of transcription factor proteins called activators. The activator proteins are not shown.
So come to a very important term. What is template encoding strand? So different with DNA replication, which is happen in both strands of the DNA template. The transcription happen only in one template. So in here, the same with the DNA polymerase, the RNA polymerase only working on a polymerase only working from 5' to 3'. Okay, so as a consequences, first it will binding into the other strand. This strand here, the 3' to 5' here. So the RNA polymerase will binding in here and start to synthesize the mRNA. So because the transcription factor binding in this template, so we call it the template strand, because the transcription factor use it as a template. And following the complementary Chagat rule, we will have A will link to U. So the second is the new mRNA will be U binding to A, C binding to G, A is binding to T, G is binding to C, and so on. So this sequence, the sequence of this will have the same sequence like the other strand of DNA. So the sequence of this mRNA will have the same sequence as this strand. So replace only the T by the U, all other sequences are the same. So that is why we call it the DNA coding strand, because the mRNA have the same sequence with this DNA strand. And sometimes we call it also the positive strand, and the other, the template strand, is called the negative strand. Okay. So I have made a small table here to compare between the differences between replication and transcription. So in DNA replication, we have template is double strand. Both strand is a template, and in transcription is a single strand. Only one strand is used as template. And as a result, it creates a double strand DNA in replication, but only one strand of RNA in transcription. Okay. The substrate, of course, this product is DNA, so this is delta nucleotide. TNTP, TP is stand for 346, and in here is the ribose nucleotide 346. Okay. In DNA replication, it requires RNA primer, but in transcription, it requires no primer. So you can see it here. There's no primer in here. The RNA polymerase can synthesize directly from the DNA template without any primer. And the enzyme one is DNA polymerase to provide DNA, and the other is RNA polymerase. Okay. So I think today's lecture is finished here. The next part on the gene structure of the prokaryotic cell and the eukaryotic cell, we will discuss about it in Monday. Just one thing before you sign up this class is that in the Moodle account, so I have updated the slide for the chapter 1. So if you want to keep your slide update to the lecture, please download the new version of chapter 1. And about the other chapter, I will keep being updated after I finish each chapter. Okay. Okay. Do you have any question or comment on today's lecture? Okay, so since you have no more question or comment, thank you very much for your attendance and see you on next Monday. Thank you.
Goodbye, teacher.
See you.
Goodbye, Professor.
Goodbye,
Professor.
Professor.
Thank you.
Goodbye, teacher.
See you.
Teacher.